Hello all, welcome to NEET World. Actually today the first class is basic maths, right? So but due to some time issue, time classes, we are starting the first class is zoology, later maths will be done, right? So online, am I audible? Is my voice is clear, right? Yes, I hope my voice is clear. Okay, we'll start the first class of this today. So today we are starting a new chapter, that is neural control and coordination. So this is also very important chapter. From this you will get maximum 3 questions, right? Maximum 3 questions, but minimum sometimes they will give only 2 questions only. So in this biology part we need to focus on each and every question, then only you will be on safer side, right? So we will discuss each and every topic, each and every aspect of all these things, neural control and coordination. So first of all we need to know why we are calling it as coordination. What is the meaning of this coordination? Initial in first class what I said, there are 2 kinds of cells, one kind of cell performing all functions on its own. Another group of cells, these are called as multicellular cells, which can depend upon each other cell, right? There are 2 cells, right? There are 2 cells. This unicellular cell is doing all the functions on its own, whereas this cell which is called as multicellular cells, to do one function it relies on other cells, right? So likewise we are the higher animals, we are human beings, higher organization kind of thing. So to perform one function, each and every cell or each and every tissue has to communicate with each other. So that communication is called as what? Coordination. The process of interacting of 2 or more different organs or tissues to perform one function. For example, I was doing some physical exercises or I was teaching. So when I was teaching, my nervous system is working, my muscular system is working, my sensory organs are working, all different parts are also working, right? So to communicating, to teaching, there are various systems, there are various tissues are coordinating simultaneously. Likewise, when we do physical exercise, what it will do, when more amount of physical exercise we are doing, so heart rate will be increased, then we can inspire more amount of air, so respiration system will be affected. So what happens, heart rate is increasing, so the blood circulation will be increased, right? So respiration, circulatory system, and whenever the blood flow is increased, it will affect the glomerular filtration rate, kidney system will be increased, right? So to doing one function such as physical exercise, there are a lot of systems are involved in that particular thing. Likewise, so to perform one similar kind of function, our body has to communicate with each other, our tissues or organs has to communicate with each other to do one function. So that's why that process is called as what? Coordination. Why we are learning this coordination in this neural? Because in our body there are 2 systems, one is neural system, another one is endocrine system. These 2 systems are very important for these coordinations. These neural system and endocrine system jointly together performs all the activities of the body. So whenever you learn this endocrine system, we learn hormones, right? So these are the chemical messengers which travels all over the body, and which reach the target tissues or cells also, which reach each and every place of our body, right? So while reaching each and every place of our body, it's doing its function. So hormones also, and for the transport of all these things we require the neural control, right? So this neural and endocrine, these 2 systems are the very important systems in what? Coordination, right? So that's why we are learning this neural control and coordination, very important chapter. Understand why it is coordination, why we are learning neural and endocrine, this thing, right? Done. Now the first thing is, in this neural control or neural system, what are the things we have to learn? So in this system, this neural system is made up of 2 things. One is neurons, another one is this thing is called as microglial cells, or you can call as non-conducting cells. There are two cells which is made up of different kind of cells. One is what? Conducting cells. Another one is what? Non-conducting cells. These are the two cells which plays an important role in nervous system. This nervous system is made up of two different kind of cells. One is conducting cells and non-conducting cells. What are conducting cells? Neurons are the conducting cells. These are the structural and functional unit of nervous system. And what are non-conducting cells? These are the different kind of cells which plays an important role in what? Protection. Which plays an important role in what? Protection. Not involvement in this conducting system, only protection or helping this neural system. In that only these non-conducting cells are going to function. Right? Understand that we have two different kind of cells. One is neurons, another one is some protecting cells. We will discuss all these things. Just remember conducting cells and non-conducting cells. And the first thing we have to know, from which tissue or which layer this neuron system or these neurons are coming to arise. So we already discussed in connective tissues or epithelial cells. This epithelial cells is derived from three layers of germ layers, whereas connective tissue is mesoderm, and also this muscular tissue also mesoderm. Whereas this nervous tissue, if you see, this is also coming from this. This is nervous system is arranged from this ectoderm. From this ectoderm, we have two cells. One is epithelial cells, which means the skin. The skin, best example is skin and neural system. These two systems are arising from this ectodermal layer. You see this, it includes what? Brain, spinal cord and nerves. So the nervous system is including brain, spinal cord and nerves. We will discuss after the structure and function of nervous system. We will go for the human brain systems. Autonomous nervous system, parasympathetic, all different kind of system we will discuss. First of all, we will discuss only the neurons, how the neurons look like, what is neuron, what is the structure of neuron, what it will doing, that thing first we will discuss, right. So the nervous tissue is derived from what? Ectoderm, and this tissue is made up of two cells. One is neuron cells and the one is neuroglia. So neurons are what? Conducting cells. What is meant by conducting? They transfer the nerve impulses from one neuron to another neuron. That is called what? Conducting cells. Whereas non-conducting cells are nothing but they are not involved in any transfer of information. They are not involved in any transfer of information, just only involved in protection or supporting cells in the nothing but protection. Sir, how we are protecting? See, these are I mentioned few examples. One is astrocyte, oligocyte, microglia, ependymal cells, Schwann cells, satellite cells. We will discuss all the functions. These are not involved in any transfer of information. These are only involved in protection. Understand? So first of all, we have to know what is this neuron, how the neuron look like, right? So starting with neuron, we have the center point called nucleus. So this, the starting one, if you observe, we have this body. The body of this neuron is called as what? Soma, right? So we'll draw the first we'll draw the diagram, then we will label everything, right? So this is what? This is this is branched dendrites. So from here we have this axon, long branched. So on this, what is there? There are some kind of cells. So the neuron is looking like this. So we need to identify what are the different parts of this neuron, and we need to know what are the labelings. The first one, the neuron was classified into two parts. The first one is body, or we can call this thing as soma, or we have different names, cyton, and we have another name, perikaryon. Right, all these names are implying what? This cell body, the this is the cytoplasm, this is the cell body, right? And these are what? These are short unbranched structures which are called as what? Dendrites. A short unbranched, remember these are not long branch. What is what? Long branch, that is what? Axon. Long unbranched is what? Axon. Short branched is what? Dendrites, right? So these are arising from the cell body. So here we have a very interesting, important, sensitive area. This is called as axon hillock. What it is doing? Axon hillock taking all the impulse from these dendrites and sending this impulse through this axon. It will transfer the impulse through this axon. It takes all the impulses from these dendrites and all kind of sites. It will take all the impulses from all directions and it will send through this axon. That is the function of axon hillock, center point of this neuron system, neuron. And you can see there are some cells. This is cytoplasm, right? We have some cell organelles. So we have some endoplasmic reticulum, that may be rough endoplasmic reticulum, smooth endoplasmic reticulum, right? And we have ribosomes, and we have some protein factories like endoplasmic reticulum, ribosomes, and Golgi complex, right? So all these are what? Cell organelles. So whenever you see these endoplasmic reticulum, ribosome, Golgi complex responsible for what? Protein synthesis. Responsible for what? Protein synthesis. Sir, what are the proteins which are forming in the cytoplasm? Those are neurotransmitters, right? In neurons we have neurotransmitters, very important for the nerve impulse transmission, right? So the neurotransmitters in this neuron, where they are synthesizing in cell body, very important one. Neurotransmitters, the proteins which are very important, they are synthesizing on where? Cell body only. And those proteins are present in cell body in form of what granules? Nissl granules. I draw with another color. So these are present in only in the cell body. So those are called as what? Nissl granules. That was the cytoplasm of the cell body. Here we have axon. This axon also having some cytoplasm. That cytoplasm is called as what? Axoplasm. And the layer on the membrane of this axon is called as axolemma, right? But the difference, if you see, the same cytoplasm is continued with the axon, but here there will be no Nissl granules. Why? This entire axon is responsible only for conduction, only for transmission of nerve impulse. Here we don't require any protein synthesis, only transmission of nerve impulse, transmission of this neurotransmitters only. So that's why here there will be no Nissl granules, only the conduction will be takes place. Here only the Nissl granules will be present, right? Next one, if you observe, I draw some different kind of structure on this axon. What it is? This is one shell which is called as Schwann shell. These are very important to produce one layer which is called as myelin sheath. Sir, why we require myelin sheath? Why it is responsible? Because this nerve impulse is transferred from this area to this area, right? It takes some amount of time. To increase the speed, we required myelin sheath. We will discuss when we are discussing transfer of nerve impulse, how from this area to this area this impulse is going, right? So to transfer fast, what happens, there is a protective layer or there is a conducting layer which is secreted on this cell. So that layer is called as what? Myelin sheath, which is responsible for the fast conduction. What? Which is responsible for the fast conduction. What happens then? From here to the neuron signal will jump to here. Then what happens from here to here? From here to here. So you can see there are some junctions. These junctions which are not having any myelin sheath layer, those junctions are called as what? Node of Runway. Node of Runway. The junctions in between two cells, two swan shells, where there is no myelin sheath, that junction, that area is called as what? Node of Runways, right? So that thing also we covered. And after this, what happens? Here also there is a one branch. So that thing is called as axon terminal. Axon terminal. In this axon terminal, finally I drawed one dark color thing which is called as what? Synaptic knob. What is synaptic knob? Here I mentioned all the neurons are, all the cell body is having nissl granules which are called proteins, and they are transferred along with axon and they are situated in this synaptic knob. From this synaptic knob only these neurotransmitters is going to release to the another neuron. So all the synapses, all the chemical transmission happens where? In synaptic knob. So this was the clear structure of the neuron, starting with the dendrites. So these are the dendrites which receives what? Signals from the other neurons, and these are the areas which transmits the signal to the other neuron or other gland or other muscular. So based on that, we have three different kind of junctions. For example, first one is neuron. If it is dendrite, this dendrite is also part of neuron, so that's why that junction is called as neuronal junction, right? So this is presynaptic part and this is one postsynaptic part. The presynaptic, definitely it will be neuron, because from the neuron only information has to take place. So from neuron, if the information is going to another neuron, then it is called as what? Neuronal junction. So that area is called as what? Neuronal junction. And second case, this is also neuron. Here it is not neuron, it is gland. Then it is what? Neuroglandular junction. We have good example of pituitary gland. Right, from neurons it is connected to gland, neuroglandular junction. And if it is, and third case, here it is neuron, it is not gland, here it is muscle. This thing is called as neuromuscular junction. Based on the type of postsynaptic thing, we have classified three things. If it is neuron, it is neuronal, and if it is gland, neuroglandular, if it is muscle, neuromuscular junction. So the thing is, the common thing is the presynaptic is definitely neuron, because the nerve impulse has to transfer from neuron to other cell, right? So that information, that transmission exclusively takes place in this synaptic knob. We will discuss how the synaptic knob and this synapse is going to take place. So understand the structure of neuron. So this was the structure of neuron, having cell body and axon axon terminal, swan shells, myelin sheath, axon lemma we discussed, soma, different names of soma, dendrites and nissl granules, right? So this was the structure of neuron. So we will see. So neurons are the functional unit of nervous tissue, you know, and these are electrically excitable cells which receive, initiate and conduct, which is also called as transmit impulses. So this neuron will receive the signal and initiate the signal, which means this axon hillock will be initiating that signal. Initiating in the sense of what? It will transmit the signal along this axon and it will release the neurotransmitter, which means it is conducting the impulse from one neuron to another neuron. It is taking the information, it is sending the information. That was they are telling, which receives, initiate and conduct the impulse that are done by that what? This neuron tissue, nervous tissue. And when a neuron is stimulated, an electrical disturbance, which is also called as action potential, is generated due to the formation of series of action potential which swiftly travels along with the plasma membrane. This thing we will discuss in transmission of nerve impulse. So what happens whenever the transmission of nerve impulse means there will be a generation of action potential here starting. So this action potential will be traveled along this neuron and it will reach to another neuron. That thing we will discuss, right? A neuron usually contains cell body and also many dendrites and a single axon. We already discussed one cell body and we have many one or more dendrites and also one single axon. So based on this number of presence of this cell body and number of presence of these dendrites, the neuron was classified into 3 different categories. We'll see. One is what? Bipolar neuron, another one is unipolar, another one is multipolar neuron, right? If you see, this is what cell body. From this cell body, one axon and one dendrite. If it is arising from the cell body, one axon terminals is coming and one dendritic terminal is coming, then it is called as what? Bipolar neuron, right? So for example, there is another thing which is called as one cell body, only one axon. It is called as unipolar. Sometimes what happens, this axon suddenly it differentiated into 2, one axon and one dendritic thing. Then it is what? Pseudo unipolar neurons, right? Pseudo means what? False. Initially it was happens like unipolar neuron. It is having one cell body, it is having one dendritic kind of thing. But what happens suddenly, this branch is going to cleave into 2 different areas. One is forming dendrites, another one is forming this axon terminal. So what happens, suddenly it was formed, right? So that's why we are calling that type of neurons as what? Pseudo unipolar neurons. Another one which is most important, what is multipolar neurons? One body, many dendrites and one single axon. That kind of thing is called as multipolar neurons. Here you need to remember where these bipolar neurons, multipolar neurons, pseudo unipolar neurons are located, right? For example, if you want to remember this multipolar neuron, if you see this cell body having what? Many dendrites, which means which receiving many signals, right? So simultaneously it's receiving signals from all different areas and it transferring along this axon. So which means the brain is the one having all the connections to all the body. It receives all the signals from eye, ear, hands, limbs, all these things, right? So definitely this multipolar neuron, they are present in where? In our body or multipolar neurons. Example we didn't give it. Brain. Brain is having this multipolar neurons, right? And also this bipolar neurons having different areas, so it is receiving from one direction and it is sending in one direction. So these are situated in this afferent and efferent nerves. So the connection where the central nervous system and body was connected, so those connections are what? Bipolar neurons. Exclusively brain is made up of multipolar neurons. Like that we need to remember all these things. So these are some matter which was I given in this slide. See, we'll discuss cell body. Cell body is also called as what? Perikaryon. We already discussed cyton and soma. And the cell body contains cytoplasm, which is also called as granular cytoplasm. Why we are calling granular cytoplasm? Because there are some granules kind of thing which are called as Nissl granules, responsible for what? Protein synthesis. So that's that's why this cytoplasm is called as granular cytoplasm. Contains very important one, it is called as what? Nissl bodies. They represent, Nissl bodies are nothing but what? Proteins. Whenever the proteins are synthesizing means there should be protein machinery. What is the protein machinery? RER, rough endoplasmic reticulum, and all the Golgi complex and this ribosomes, all are coming under this protein synthesis machinery. and a group of cell bodies in the central nervous system is called as nucleus, and if it is peripheral nervous system, it's called as ganglia. There are two words given in this book. So they are telling that there are group of nervous tissues which is present in central nervous system. That group of tissue is called as nucleus, and if it is peripheral nervous system, ganglion. We will discuss what is central nervous system, what is peripheral nervous system. In that, the central nervous system neurons are called nucleus, whereas peripheral nervous system neurons are called as what? Ganglia. So that was the part of cell body. Coming to the dendrites, see, these are short, branched, which are arising from the cell body, is called as what? Dendrites. From the cell body only one long unbranched kind of thing which is arising, that is called as what? Axon. Right. So these are the dendrites. What are they doing? They conduct nerve impulse towards the cell body, which means it receiving the signal and it is giving to the cell body. See, towards the cell body. Understand? So here you need to remember there are two kinds of nerves. One is afferent and efferent nerve, that we will be discussing nervous system. So there will be two kind of nerve. What is afferent and efferent? Same kind of thing we will discuss in excretion also. Afferent neurons and efferent arterioles. That can be same thing, which is entry and what is exit. Same thing, right? So that was the diagram. So axon. Axon is a single long cylindrical process, that is called axon. This axon has one plasma membrane which is also called as axolemma, and the cytoplasm is called as what? Axoplasm. As I mentioned here, the nissile bodies are absent, right? So this axon is along and it is finally reached at where? Telodendria, which is nothing but the axon terminals. Axon terminals also called as what? Telodendria. At the last part, we have some bulb-like structure, and that bulb is called as synaptic knobs or terminal buttons. Here what happens? All the neurotransmitters, which are proteins, are situated in some vesicles in this synaptic knob. So those vesicles are called as what? Synaptic vesicles, which contains what? Neurotransmitters, which are very important for this nerve impulse transmission. They are situated in this synaptic knob, right? So what this axon is doing, it is transferring the nerve impulse from one neuron to another neuron. It may be interneuronal, it may be neuromuscular, or it may be neuroglandular also. That thing also done. Next one, see, this was the structure of neuron. So these are the dendrites, all the nucleus was there, and the protein machinery is also there, the cytoplasm. And here we know, right, axon hillock, origin of axon. From here the axon will be made, and from this here only all the impulse was received and transferred along with this. And we know, right, there are some kind of cells called Schwann cells. These are responsible for made up of this myelin sheath, and we know these gaps are called as node of Runway, right? See, the axon, the layer of this axon is called as axolemma, and the cytoplasm is called as axoplasm. So that was the thing. So the final synaptic knob, when I zoom, this was one synaptic knob. This synaptic knob contains small vesicles. These vesicles are called as what? Synaptic vesicles, which contain small granules. What are these granules? Very important. These are called as what? Neurotransmitter. The best example of neurotransmitter is acetylcholine, right? So see, this is one neuron. This is presynaptic neuron, and this is what? Postsynaptic neuron. In between we have space, this called as cleft, which is also called as synaptic cleft. We will discuss detail in synapses. We have two types of synapse, one is electrical and the one is chemical synapse. In that we will discuss, right? So this is about neuron, right? So based on presence or absence of myelin sheath, right? I said there is one cell which is very important, which is called a Schwann cell. This Schwann cell is for conduction of this fast conduction of the neuron. So this Schwann cell is producing one layer which is called as what? Myelin sheath. If the neuron is having myelin sheath, then these neurons are called as what? Myelinated neurons. and the axons are called as what? Myelinated axons. If the neuron is not having any myelin sheath, which is it is not having any Schwann cells, it is only axon, so then that is called as non-myelinated neurons or non-myelinated axons. Understand? We have two categories. One is some neurons having Schwann cells where containing myelin sheath. So that's why those neurons are called as what? Myelinated neurons. And there are some neurons, they are not having any Schwann cells, so there is no myelin sheath, so those are called as what? Non-myelinated neurons. And we have two systems, right? One is central nervous system, another one is peripheral nervous system. See, in the central nervous system, the glial cells are called as oligodendrocytes. These are also responsible for the myelinated axons, right? We have two kind of cells. One is what? Schwann cells, another one is what? Oligodendrocytes. These are the two cells which are coming under neuroglial cells. I am telling, I already told, right, two kind of cells. One is neurons, another one is neuroglial cells. One is conducting cell, another one is non-conducting cell. These two are coming under what? Non-conducting cells, which are very important to produce myelin, right? We have two systems. One is central nervous system, another one is peripheral nervous system. In central nervous system, the cells are what? Oligodendrocytes. These are responsible for to produce myelinated axon. In peripheral nervous system, Schwann cells are present. These are responsible for the myelinated axons. In two systems, two cells. That should remember. What system, what cell is having. Peripheral nervous system, Schwann cells. Central nervous system, oligodendrocytes. Right? So that thing. And also, whenever you see this brain structure, we have two kinds of matter. One is white matter, another one is gray matter. Because of the presence of this myelin also it is coming, so that we will discuss in nervous system. So let's just revise that thing. So for example, this is the cross section of this brain, and we have some gray matter here and we have some white matter here, right? So what kind of neurons are present, or what part of neuron is present here, and here what part of neuron is present. So for example, when I draw again the neuron, all the dendrites and the axon and telodendrites. Here if you see, this portion is having what? Myelinated portion, whereas this portion is not having any myelinated portion, which means whenever the cell is having myelinated neurons, which gives one color which is called as white, which means the white matter, the area, the brain which is having white matter are exclusively presence of this myelinated axon portion. In this neuron, all in this brain, all the thing is made up of what? Axon portion only. Whereas in gray matter, what is there? Only cell body. The part of the neuron, cell body is having in gray matter, and this axon is having in white matter. So here white matter, white color is due to presence of this myelin sheath. Just remember, myelin sheath is white color. This myelin layer is white color. That's why this part is present in white matter, whereas this cell body does not having any myelin sheath, so no color, so that will be gray matter. So this thing we will discuss again in nervous tissue also, this systems, right? So finally we discussed all the parts of neurons, what is called conducting cells, and we have another type of cells which are called as what? Supporting cells, also called as what? Non-conducting cells, right? Supporting cells are also called as what? Non-conducting cells. What are non-conducting cells? The first one is oligodendrocytes. So this non-conducting cells also classified into two. One is what? Based on the location. If these non-conducting cells are responsible for the central nervous system, so this is one part, and if it is peripheral nervous system, this is second part. So non-conducting cells classified into two based on the presence of systems. So the cells which are responsible for the central nervous system are four. What is that? First one is oligodendrocytes. We already discussed about this. Oligodendrocytes are responsible to produce myelin sheath, on the neurons. So which causing myelinated neurons in where? Central nervous system. And next one is astrocytes. Astrocytes are nothing but what? Star shaped cells. that form interconnection between the neurons. So we have two neurons, this is one neuron, this is one neuron. So for example, this is the one axon of neuron, this is also one axon of neuron. So in between there is a star shaped cell which is responsible to hold or to communicate between two neurons. So those are called as what? Astrocytes. And there are another kind of cells which are called as ependymal cells. So this thing, it's a modified epithelial cell. So we already discussed epithelial tissues have two modified things. One is kind of cilia, another one is called microvilli. Likewise, these brain cells or ependymal cells are also some modified cells which are having what? Ciliary kind of thing. These are present in where? Lining the cavities of the brain, spinal cord, to bring the movements in this cerebrospinal fluid. And the final one is very important, microglial cells. These are also called as macrophages. We already discussed monocytes are transformed into these macrophages whenever they entered into tissue. If the monocyte is entered into the brain cell and becoming macrophage, that macrophage is called as what? Microglial cells. What is the function of this microglial cells? Whenever there is a pathogen, whenever there is a debris is present in this brain, then it is going and engulf that particular cells. Phagocytosis will be happen. So that kind of thing was done by this macrophage. What is the name of this macrophage? Microglial cells. Very important. 4 cells are included in central nervous system, and there are 2 cells. One is what? This thing, satellite cells and Schwann cells. Right? Schwann cells we already know, these are responsible for this myelin sheath. Right? Satellite cells surrounds the cell body in ganglia. We already seen 2 kind of thing, one is ganglia, another one is nucleus. Central nervous system neurons are called as nucleus, whereas peripheral nervous system neurons are called as what? Ganglia. So whenever the peripheral nervous system ganglia is there, that ganglia is surrounded by what? Satellite cells for the communication only. So if you see the diagram representation, if you see oligodendrocytes, so these are oligodendrocytes forming what? Myelin sheath. And these are what? Astrocytes. Astrocytes present in between one neuron to another neuron. Here they are showing capillary. The connection between 2 things, capillary and neuron. The connection will be appeared by this astrocytes. And if you see the layer of the cells having some modification which is called as ciliary modification, responsible for what? Bringing movements in cerebrospinal fluid. And there is another kind of cells, microglial cells. Whenever there is a debris, whenever there is a pathogen, whenever there is a foreign body which is attacked in this tissue, then what happens? This cell is going to engulf or destroy this pathogen. That is called macrophage, phagocytosis. That is done by microglial cells. And also we know red Schwann cells. On the axon, we have one cell which is called a Schwann cell responsible for the myelinated thing. And also there is one cell called a satellite cell responsible for the providing support and structural support, functional and support to the particular ganglia. So these are the different kind of cells which are involved in what? Protection and support to the tissue. What tissue? Neural tissue. And there are another kind of cell, that is neuron. So this neuron and this non-conducting cell together we are calling it as what? Neural tissue. Right? So starting we already discussed, there are some group of cells together to perform one function, and that group of cells is called as what? Tissue. That may be same group of cells, that may be different group of cells. We discussed the general equation or the general statement is that may be same group of cells together to perform same different kind of function is tissue. But here if you see in nervous tissue, the nervous tissue is made up of different types of cells which are having different functions, but ultimately the function is what? Nerve impulse transmission. Though it is showing nerve impulse transmission function, but individual cells having different functions. So different group of cells having different functions, but ultimately having one function. So that group of cells performing one tissue is called as nervous tissue. Understand the nervous tissue? It is made up of different kind of cells, right? right? So we need to know how the transmission of nerve impulse is going to take place. So this was the last part, so we'll see. So this is also one neuron and this is another neuron. So this is area, the gap between the two neurons, and this is the area of the neuron. For example, there is a one transmission was happen, there is a nerve impulse generation was happen, and all the signal was received by this dendrite. Now what happens? This dendrite receives the signal and it will initiate the signal along this axon, and the signal and released or reached to the synaptic knobs. Now what happens? There is a gap between this one neuron and another neuron, and this gap, what happens? There will be signal has to trace, the signal has to transfer, but here there is a gap. So in this gap there will be next another kind of molecules that will replace the function of this transfer of this nerve impulse. So with the help of some chemicals, some chemicals or proteins, we are replacing the electric signal which is coming from this axon hillock to this synaptic knob. With the help of these chemicals, we are transferring this information to one neuron to another neuron. Likewise, in this area, we are transferring the information in form of electric signal, right? Here in form of chemical signal, and again electric signal, again chemical signal. So the information or the depolarization, in scientific words, depolarization or the information in this nervous system was transferred by two kind of methods. One is electric signal method, another one is chemical signal method. We need to know two things also: how the electric signals are transferring along this neuron, and how the chemical signal is transferring in between neuron. Two words: along with the neuron, in between the neuron. Along the neuron, the signal is transformed electrically. In between neuron, the signal is transformed chemically. We know this is neurotransmitter. Now we will see how action potential is generated, how one neuron is getting excited and transfer the nerve impulse transmission, right? We will see. So this is axon. This axon having some ions and some proteins. We will see what are the different kind of proteins. Inside of axon, we have potassium ions and also some negatively charged proteins. Outside of the ion, we have what? Sodium, right? So if you see here, K plus is there and more amount of this negatively charged proteins are there. So initially in resting membrane, which is nothing but in a normal state, resting state, the nerve impulse inside is what? Negative, because there is more number of negative charged proteins. And outside, if you see, there is more amount of sodium, so that's why here it is positive. This condition is present in where? Resting membrane potential. Inside is negative due to K plus and positively charged proteins. Outside is positive due to Na plus. This is in resting membrane potential, right? Resting membrane potential. So if you calculate the voltage inside, you will get minus 70 millivolts. That was the number of the resting membrane potential, minus 70 millivolts. Now what happens? Whenever there is a signal was generated, whenever there is a signal was transferred or taken up by this axon, what happens? There is a change in this ions, there is a change in this charge balance. What happens? We will see. Let's come to down. Okay, here only. Now what happens whenever there is a initial signal was generated? What happens? There are some channels, right? This thing. So on the membrane we have some channels for sodium also, we have some channels for potassium also, and we have some channels for these channels are called as leaky channels. These channels are called as what? Leaky channels. Along with channels we have some pumps. These are called as what? Sodium potassium pumps. Initially I told there is a resting membrane potential, inside is negative, outside is positive, and the negative charge is minus 70 millivolts, right? So this is all with the help of this sodium potassium leaky channels and sodium potassium pumps. Later you will understand. First what you need to understand, how the action potential is generated. Whenever there is a generation of this action potential, what happens? This opening of sodium channels will be done. The first one is opening of sodium channels will be done. Whenever there is a open, what happens? We have outside more Na plus, right? Outside more Na plus. What happens? All the Na plus will be entered into inside. All the Na plus will be entered into inside. Then what happens? Inside will become what? Positive. Inside will become what? Positive. What happens outside? Outside will become negative. Resting membrane potential, here inside there is K plus and negatively charged ions, and here outside Na plus. After what happens? There is a opening of sodium ions, sodium channels. What happens? All the sodium ions was entered from outside to inside. Then what happens? Inside there will be more positive. All the plus sign was here, more positive was there, and outside there will be low Na plus, so that is negative. After some time again there is now the sodium channels will be closed. After some time what happens? Potassium channels will open. We know that inside potassium channels are red, so they will go out. The first step, opening of sodium ions. Second step, opening of potassium ions. Whenever there is an opening of potassium ions, all the K plus was go into outside. Now what happens? Again it will become negative. Here it will become positive. Why? There will be more amount of K plus outside, there will be low amount of Na plus inside. If you see these two, both the charges are same. Outside negative, outside positive, inside negative, outside positive, inside negative. But the difference is, here negative is due to the K plus and uncharged this thing. But here negative is due to Na plus. Here it is due to Na plus, here it is due to K plus, right? For that we need to bring back these ions into the same position, like this position. For that we have what? Sodium potassium pumps. Just remember, sodium, Na plus, potassium, K plus, Na plus 1 2 3, K plus 1 2, which means for every 3 sodium ions there will be a replacement of 2 potassium ions. 3 sodium, 2 potassium. Just remember 1 2 3, 3 sodium, K plus 1 2, 2 potassium ions. So here what we required? So here we have Na inside, K plus outside. We need to bring all the Na outside, and we need to take all the K plus inside, right? So for every 3 sodium ions there will be 2 potassium ions will be go inside. Likewise sodium potassium pump will be done, and again this phase is going to coming into resting membrane potential. Here what happens? So we know, right, physics. So the charge transfer will be happen from positive to negative. So likewise whenever there is a positive happens out that side also, here it is negative, right, this side. So this charge will be go into positive to negative, and this charge will come to this side. So there is a charge transfer will happen. So if it is neuron, the first portion is depolarized, and after that what happens? Remaining side there will be a kind of negativity. Right? For example, this was confused. So this is one. We have 2 portions. The first portion is resting membrane potential, which is negative, and outside is positive. What happens? Depolarization, plus was inside, negative one's inside. So along with this is also positive, right? All this neuron is positive, this is also plus. All the neuron inside is negative, outside is positive. Only this portion, what happens? Depolarization happens. All the along the neuron is negative. What happens? This charge will go to this place and this charge will come to this place. Now what happens? This portion was depolarized. Next portion, this portion will be depolarized. After that, same thing will be happen. Next what happens? This portion to this portion will be depolarized. Likewise, one portion, if it is depolarized, along the neuron, along the axon, the particular portions will be depolarized. This is how the transmission will be happen because of the electrical negatively positively charged. The plus always attracts this negative, or this negative was attracted by this plus. So whenever there is a depolarization happens, inside will become positive. This positive ions all will be attracted towards this negative side, and outside all will be attracted to this positive side. Now this portion of the neuron depolarized, this depolarization shift into next portion, and later also now all this become positive and here all will become negative, and this again plus will be shifted to the next portion. Likewise, one portion of the neuron or one portion of the axon will shift into next portion. In this diagram they clearly mentioned. See, starting is positive and this positively charged was transferred into the negative side. See, the positively charged are transferred into the negative side and positively charged transferred into this negative side. Likewise, this portion is transferred into now another portion of this axon. Again depolarization takes place here. Now depolarization was shifted into this portion, and again depolarization was shifted into this portion. That's how from starting of the neuron the signal is transferring into the ending of the neuron. Understand how that signal was transferred, right? Understand, it's very clear to understand. Because of the charge reversals, all the information is going. Why the charge reversals are happening? Because of the ions. Why ions are coming? Because we have one thing which is called as depolarization, opening of the sodium ions. Why the opening of the sodium ions are doing? Whenever the action potential is generated or coming, this sodium ions will open. All the sodium ions will rush into the axon. Then there will be a positive. This positive will attracted by this side. Now what happens? This we have to again the charge reversal balance by ions. Ion charge will be automatically reversed, and this positive sign ionic balance also will be reversed. All this thing will be come into done by this sodium potassium pump, and we have some channels called K plus channels, and we have some channels called Na plus channels, and we have some channels called leaky plus channels. All this thing plays an important role in this conduction of nerve impulse. All will do simultaneously work. Understand, that was the transmission of nerve impulse, or transmission or conduction of nerve impulse, right? Understand. The first one is depolarization. Just remember, depolarization is nothing but entry of sodium ions into the cell. That causes definitely depolarization. That may be a neuron, that may be a muscle, or that may be in any kind of thing. In heart also we have depolarization action potential. Wherever, whenever you see there is a word called depolarization, that means entry of sodium ions into the cell, into the cytoplasm. That causes what? Depolarization. Depolarization is nothing but what? Generation of action potential, generation of signal. Whenever the signal was generated, the signal was shifted to next place of the neuron, next place of the neuron. That's how the signal was transformed to transferred to one portion to another portion. For example, if it is non-myelinated neuron, the signal will go like this. If it is myelinated neuron, which means here we have nervous myelin sheath, which is also called as this Schwann shell, then what happens? From this portion directly it will jump into next portion. That's why myelin sheaths conducts fast nerve impulse transmission. From here to here it will take generally 5 minutes, means if this portion is not involved in this neuron nervous transmission, then it will take 1 minute, right. So that's how myelin sheaths are responsible for the fast impulse transmission, right. So today we discussed the nerve impulse transmission also, and we discussed the structure of neuron, and we discussed some different kinds of cells which are helping in the protection of nervous tissue, right. So clear. And we will end this session. We will continue tomorrow class how the synaptic transmission will happen. Later we will see the parts of brain, right. Okay sir, okay. Thank you. If there are any doubts, we will discuss in next class. Thank you so much.